Coming to class on this beautiful. All right, let's get started. Thank you for coming to class on this beautiful day. I really think this should help you develop a sense of your interior neuron and this functional simulation you have. I hope that you all are building a movie where you can zoom in and imagine neurons talking to each other. And so we're trying to get you to draw and think about neurons and circuits working together to pass information, so we can go from the sensory inputs out to the motor outputs, generating the correct behaviors for the situation that any individual is in. All right, and that information can change with learning. And so we're going to talk about neurotransmission at the level of one neuron talking to another today, but we're going to put it in a behavioral context that we developed last week. All right, so remember we talked about PTSD and fear behaviors that can be very adaptive, but can also be maladaptive in a modern setting. For example, we could have a veteran who returned from a theater of war and is now trying to go to college and have a more peaceful life. They might be in a nightclub and hear a very loud noise, which could cause a fear response, or it could be something that they ignore and they go on with their day. So we can imagine that this a loud sound that could potentially be a gunshot, or it could be a balloon pop or some kind of foam cannon or something like that that's harmless. And we could place this in a circuit. We've talked a lot about the amygdala over the weeks here, and we've talked a lot about fear conditioning. And so you've seen the spaghetti here of the amygdala, and you know that the red pathways here are one of the fastest ways that a sound could travel through this threat learning system and generate a fear response. So this pathway here, fear neurons projecting to the central nucleus, can generate a fear response. Of freezing is something that's studied a lot in rodents. Freezing, arousal of the whole body, autonomic arousal, which we'll talk about more in a few weeks. These could be fear responses that could also happen in a person who's in a nightclub situation, and it could ruin their night. All right. And also drill neurons. Their individual cell bodies aren't drawn here yet, but you can imagine that they have GABA and glutamate inputs onto them. And we know from this circuit that the extinction pathway, one output arm of this long extinction pathway, is a GABAergic input onto the central nucleus. And we know that one output of the fear threat pathway, this fear learning pathway, is a glutamatergic input onto the central nucleus neurons. And so it's not always the case that GABA and glutamate are labeled one extinction, one fear. There could be other synapses and other places in the brain where the fear pathway is using GABAergic inputs, and the extinction pathway is using glutamatergic inputs. So this is not a hard segregation at all sites. But at this one particular site, this is what's happening, and they could be in opposition to each other, driving a potential fear response or shutting it down. And so this I'm trying to get you to have a sense of a stake of what's happening, whether or not this central nucleus neurons in this location are going to fire or not. And just so you follow, sometimes glutamatergic neurons are drawn with this kind of symbol. Here they're indicated by a red arrow. And this is another way of indicating an excitatory synapse. And this ball is a way of indicating an inhibitory synapse. It's just a little notation neuroscientists use. So this is a little legend to help you with that notation. GABAergic and glutamatergic. Everybody's with me here? So we're in a location in the brain. We have competing inputs, and there's something at stake here. Okay. All right, and so let's place a neuron there. You can see here there's some big glutamatergic inputs with that notation flip I made here. So you can imagine them synapsing as presynaptic terminals onto the dendrites of the central nucleus neuron. And here's a GABAergic synapse coming onto those same dendrites. So then we get this competition to either drive or stop a fear response. Any questions? If anyone's lost, this is, hopefully you're localized now. Yes? In reality, how how many neurons can be sufficient for generating a behavior? That's a great question. Tens of neurons can be sufficient for a behavior. Even though the brain is pretty vast with so many neurons, you can use optogenetics to do experiments with just 10 neurons, and you can find that small a number can be sufficient to generate a behavior. It's a different context, but that's kind of a lowest number that we could see works. Yeah. Okay. And so now we've got this location, we've got something at stake at the behavioral level. Now hopefully you're motivated to follow me into the interior of the neuron, to understand what happens in the interior of the neuron, to find out the end of this story. All right, and so we're putting an electrode, a glass electrode, into the inside of a neuron, so we can eavesdrop on the interactions it's having with other cells through changes in electrical currents. And there are ions on either side of the membrane of the sac, which is the outside of the neuron. The neuron is a big lipid sac. And because the charges are not balanced on the two sides, this neuron is alive and it's sitting at rest. One of the features that breaks down when a brain dies is this balance of ions, not balance of ions, but this imbalance of ions across the membrane is important for life. It helps your brain work, it helps your heart work, and this is what helps us stay alive, is that there's this imbalance where there's more negative charges on the inside of the neuron and more positive charges on the outside. All right, and this difference across the membrane is called the membrane potential. It's a vocabulary word that might be new and awkward, but you can write it down, work on understanding what that is, and it'd be good to talk in section and come to office hours to talk about that. And neurons are very often firing, but we can talk about the times when they are not and think about them sitting at rest, sitting basically with this negative potential on the inside, where there's more negative ions on the inside than on the outside. And very often they're in flux and this balance is shifting, but the neurons are working to stay close to this resting membrane potential. Now I'll dig into that a little bit more. So we've set up the context of when this matters. We set up a location for this to matter, a neuron in the central nucleus. And so what's going to be important is the balance of ions across the membrane of this central nucleus neuron. And this is going to be determined by inputs to the neuron, GABAergic and glutamatergic inputs to the neuron. And then we're trying to understand is whether or not this neuron is going to fire an all or none action potential. And so you've become familiar with my teaching so far in this class. I've taught about inputs onto the dendrites, but we've spent less time talking about the all or none generation of something called an action potential. And that is going to start here at the axon hillock and propagate down the axon and release neurotransmitters in the axon. And that is going to determine whether or not the animal produces that fear response or not. The inputs are coming in on the dendrites through ligand gated a little bit. There are two glutamatergic ligand-gated receptors. Their names are AMPA and NMDA. And we will be studying more and more detail over time. Positive ions. The AMPA is a large, basically, workhorse of the central nervous system for glutamatergic neurotransmission, and it's largely letting in sodium ions. NMDA type glutamate receptors, they're also bound by the ligand glutamate, and they are more reserved for learning and plasticity, strengthening and weakening of synapses. They let in sodium as well, but they also let in calcium. Those are both positive ions. All right, so they're both excitatory. All right, GABAergic receptors are the receptors that are responsible for these inhibitory inputs we're talking about, largely in the CNS. The ligand that binds them is GABA. They also have these sites where barbiturates and alcohol can modulate how well they work, facilitating how well they work, and that's why these are considered downers, because they help to inhibit the nervous system. And this is because the ion they're letting in is chloride. All right, so more negative ions can come through this type of receptor. So this is a little bit of a refresher from a couple weeks ago. And so we've got these out on the dendrites, right? So now we have these, if you localize these out onto the dendrites, we're going to call the flux of ions through these ligand-gated receptors, these 3 ligand-gated receptors we just talked about, we're going to call these graded potentials, okay? And the word potential is about flux of ions across the membrane. It's a change in the membrane potential. That balance of positive and negative across the membrane is being affected by this flux of ions. Okay, and so these graded potentials, we're going to largely localize these events to the dendrites. And then we're going to talk about the action potential now, long-awaited action potential, and we're going to localize this all or none event and its initiation to the axon hillock. And the important part of this for thinking about neurotransmission is when it propagates. That's a new vocabulary word for some of you. It's a word we're using to describe the movement of the action potential down the axon to the terminal boutons. And I hope you can read that in the back, but hopefully you can see it on your screen. The writing is kind of small, and I've got a slightly bigger version of this diagram at the end of the slides too. It doesn't have any overlay on it. I like the big size of the dendrites on this diagram and then all of the annotation. It's not in your textbook, but it is in your slides. And so it shows that the action potential and the important transmission that occurs because the cell fires is happening because the axon undergoes an action potential which will release neurotransmitter at the terminals. And we've talked about that a little bit, where we have vesicles full of neurotransmitter which are seen to be releasing neurotransmitter into synaptic cleft here. Because an action potential has arrived at the terminal, something called voltage-gated channels that allow in calcium open here, which helps to trigger the release of neurotransmitter. And this is a quick view of this, and we're going to walk through this the rest of the hour today. Okay, so if you're a little bit lost, this hopefully is think I need to organize graded potentials out there in the dendrites and think about how they sum to create an action potential in the axon. And then finally, how does that at the end result in neurotransmitter release at a synapse at the end of the axon, at the terminal boutons? Okay, so you're going to have this 1, 2, 3 events to think about today. And just have these placeholders, and we'll fill in these issues. Okay, now the action potential is something you haven't heard about too much. Its discovery won the Nobel Prize. Here's the action potential recording made by the Nobel laureates here, Hodgkin and Huxley. There they are. And it's a little fun fact is that they used the squid to study this. And the squid has been a favorite of neuroscientists because they have this enormous axon that's so big you can stick an electrode into it. It runs through the length of the body of the squid. It can be dissected out. And it's important for jetting the squid away when something is attacking it. And so it's this all, danger. So it's a threat response for the squid, and then it can jet away. And it has its most important task in its life, I guess. And so it developed this enormous axon to help generate this event. And Hodgkin and Huxley realized this was part of the nervous system, and it was the most convenient place to study the nervous system, and stuck an electrode into this and measured this incredible event. It's very brief. It's just a few milliseconds long. It's very dramatic. It goes from negative resting potential to very resting potential and back again in milliseconds. And so it's this precise and violent event which is critical for the function of multicellular organisms with nervous system. And the violence, I mean, is, why would I say violence? And the movement of the ions is enormous and in 2 directions and very fast. So it's if you want to think, you're making, just pat yourself. You can do one of these patting things, make some action potentials. You're generating, think how fast you can generate some self-tapping here. Yes, thank you for participating. Give yourself a pat on the back. You're generating action potentials. And so these are very fast events. Neuroscientists get very, this is a real flex when you think about how slow a lot of molecular biology is. I remember being at a lecture of Rodolfo Linares, a professor at NYU, went up to the front when I was a graduate student. He said, "You molecular biologist?" And then he looked at this guy and went, and pretended to punch him. It's like, neuroscience, you know. But now I don't do the real punch. This is 30 years later. So, all right, so these things are fast. And so we're going to fill in. This is, again, just a quick guide to where we're going. We have ligand-gated receptors out here that are important for the graded potentials. And then down here, after the cell body, we're going to have the axon hillock. And stay tuned for voltage-gated sodium channels. These are the critical initiators of the action potential. Sometimes people think the action potential happens and these things are important. Well, they generate it. This is the mechanism that's going to generate it. All right, so stay tuned to see how those do it. And it's an all or none domino effect down the axon that channels are opening, that are voltage gated. And then it culminates in further opening of voltage gated calcium channels here. All right, so that's what this light traveling down a cell is in the videos you see that are animations. Okay, so let's walk through this in our central nucleus neuron, somewhat messily slapped on top of the diagram from the textbook. And here's its axon hillock labeled, and here are its inputs. And so these two inputs are competing to get this axon hillock to generate an action potential or not. All right, and I told you that there's a lot of negative ions inside a neuron. So this A- means anions. It's a vocabulary term for negative ions. That's why there's a negative there. And this is giving you the concentrations of the ions. And the interior is high in potassium. The exterior is high in sodium and chloride. So we call it the banana in the ocean. Bananas are full of potassium. And so if you can't remember which ions are on which side, it's the banana in the ocean. Okay? And so that's how it is at rest. And then during an action potential, these things move violently through the voltage-gated channels. And that's how we get an action potential. That's what makes us alive and able to generate things. And so this is a lab member I have mentioned here that he got lost a little bit when he took my class, that he was wondering about how the resting membrane potential relates to the movement of ions and all these graded and action potentials. And so there's to fire once and then to fire again, a neuron has to get back to its resting state so that it's ready to fire again. So he explicitly added this slide so that we put this in place here. The neuron, after it's fired, it needs to get the sodium potassium back on the correct side. And so it has this special thing called the sodium potassium pump that complements some natural diffusion that can happen across the membrane of the cell, where potassium can diffuse out and sodium can diffuse in. So this actually works against that diffusion grading here, that it can bring potassium in to get the banana more banana-y, right? And it can pump sodium out. And so it uses cellular energy to maintain this balance, to keep neurons ready to fire. And this is not an MCB course. We're not going to go into this in any gory detail. It's just like, how does this thing work? This gives you a little sense that there's some of the calories that you consume today were being used to keep your brain in the right shape, that it's ready to have action potentials and graded potentials. Okay. A lot of the calories that you use today are keeping the house the right state so that you can have your neurons firing. A friend of mine's mother is in the hospital because she has an electrolyte imbalance and her cells aren't keeping these things correctly, and she became very confused and had to go into the hospital, and her heart stopped working properly because the cells aren't managing this well. So it's, this is, as you run the marathon and you drink too much water or too little water or don't have enough electrolytes in your Gatorade, this would be a reason that you would maybe show neurological symptoms and need to go get properly hydrated. Okay. All right, so we have hopefully established a cell being at rest. I'll just pause there, and anyone have any questions about a cell being at rest and these ions? Want to check in with your neighbor for a second about it? Yeah, why don't we do that? Just what is it being at rest here? What is this banana in the ocean? All right, thanks everybody. Any questions? Did people hit any walls here? Any questions? Anything come up that quickly clarify to help everybody? If you're wondering about something, maybe somebody else has the same question. You sure? Yes. Yeah, it's one of the major features, and you're going to see also there's going to be a voltage-gated potassium channel that'll help too. Okay, so there's going to be a help with the movement. Okay. All right, so we have the cell sitting at rest, and we're gonna cause a big change in the neuron by release of neurotransmitter from our presynaptic terminal onto the postsynaptic spines on the dendrites. And another just point of clarification, a neuron can be both presynaptic and postsynaptic. It receives thousands of inputs, so it's postsynaptic to those presynaptic inputs. But then, if maybe the dendrites are here, the soma is where I'm standing, and the axon travels all the way over here, and synapse is on Carter's neuron, that would be, it would be presynaptic. This neuron that maybe I'm the soma of would be presynaptic to Carter's neuron, which then would have its own axon. It'd be postsynaptic, and then its own axon, and go back to, I think, where's Marnie or Alex here? Okay, does that? Can you see how in a chain of neurons, it could be both presynaptic sending outputs, and then it would be postsynaptic receiving the outputs. So there's some other neuron here that's presynaptic to me, and I'm receiving these inputs and sending them out. Okay, and so there's these long chains, of course, and they can even be reciprocal, so that you could have a loop in the brain. So how, and what's important to know here, and we'll just go quickly into some membrane biophysics, is that when there's concentration gradients, either just the chemical difference or an electrical difference, there's driving forces that will help diffuse the ions across any opening between two compartments. And so imagine two compartments in your bathtub. Let's say you were able to put a large plastic bag full of water in your bathtub, and you filled it full of red dye. And it's sitting there in your bathtub, and there's no red dye in the rest of the bathtub. Okay, and it would just be full of red dye, but if you poked a few holes in that bag, you would have more red dye here in this bag than in the bathtub, but eventually over time, because the concentration was higher in the bag than outside the bag, it would diffuse until there was equilibrium between what's in the bag and what's outside the bag in the bathtub. All right, and so the brain is kind of the same way. The bag is the neuron, and the chemical gradient, it could have greater concentration in the neuron or greater concentration outside the neuron. It's going to move, be more likely to be moving against its gradient. Okay. You can also have electrical driving force. So instead of moving from just, we imagine particles that are neutral, we have charged particles, and the charge on them is like static electricity, or it is static electricity, is made of things like this, and this charge can also help move things. It can create forces. And so you can have electrical driving force where there's more negative ions on the inside. The negative ion the force is pressing it out. And if you have more positive ions on the outside, the force will push it in. Okay. So the bag could be full of chloride, and sodium could be on the outside. All right, and so when these ion channels open, another important element here is that they don't let all ions pass identically. They can be selective for specific ions, and so they might just let chloride in, or they might just let sodium in, and so they also interact with these forces. And so you get this complex picture of chemical driving force and electrical driving force, and also the ligand-gated ion channels that open are sometimes selective pores. Their very conformation determines which ions can come in and which ions cannot come in. All right? Or they might be voltage-gated. Potential across the membrane, the charge across the membrane is conducive to their opening. All right, so they open and close. Like traps you used to get at the doctor. Your shots, they give you a toy, and they have these little woven baskets that you put on your fingers, and you can pull on them or open them, and your fingers can pop out. And so I like to think of voltage-gated channels like these traps that, depending on whether you have the springiness on them, if they're pulled tight, they're a little closed, and but if there's less force in the membrane, they can pop together. All right, so there's the voltage-gated channel. So there's a great complexity here, and you can study. This has been worked out in absolutely beautiful detail. It's probably a human accomplishment akin to decoding how DNA, RNA, protein works. The understanding of the movement of ions across membranes is captured by beautiful equations, one of them the Nernst equation. And there's beautiful biophysical experiments studying these movements, which are really important to medicine, things like cardiology. So if you want to go study that for more information or take classes in MCB, you could see a great human accomplishment in understanding these things. But we're going to stop there. Okay. So, but we will put them in context. We'll make use of this information. And we'll think about a neuron, which sits, I told you they sit at negative potentials. A lot of neurons like to sit at something called minus 70 millivolts. That's what you measure when you put an electrode on the inside of the neuron and reference it to the liquid outside the neuron. And when there's release of glutamate onto, here's glutamate, binding to two glutamate receptors, we've got positive ions coming in. They're especially coming in through the AMPA receptors. And you can see the positive ions coming in can create graded potentials, which are existing in the dendrites. These are kind of sad little dendrites in this neuron, but it was a nice diagram for the sake that it showed. The 0 line, a neuron sitting at minus 70, and then when the glutamate binds, which is happening here at time point 1, glutamate binding, it's the depolarizing stimulus. You have these positive ions coming in, making the cell less negative. Everybody with me there? Because it's sitting at minus 70, there's a lot of negative ions in there. They're canceled out by this positive sodium coming in. And so therefore there's this, it goes right near, especially this location just underneath these receptors. It's likely in a dendritic spine. This local area is more positive. okay? And these small local areas of greater positivity, which is actually less negativity, but there's a lot of positive ions there. So it's like a cloud of positive ions. These are called graded potentials, okay? And it could also be, if it was GABA that bound, it would get more negative. It'd be a cloud of more negative ions, chloride ions underneath there. That would be a negative graded potential. Okay, so everybody with me? You have this concept of clouds of ions underneath these receptors, and these are graded potentials. Yes? The whole dendritic and somatic body. So the question was, where in the neuron does the process of integration happen? And so imagine this whole body of the neuron. It can have very elaborate dendrites, and there's all these small clouds of ions flowing in. Okay, and these channels, these ligand-gated receptors with their ion channels in the inside, they're called ionotropic receptors. They're not enormous, and this is causing a local change. And so the neuron's decision to fire or not comes from integration of these ligand-gated channels all. Okay. And we're going to walk through that. Yeah. And so you're thinking ahead. How does this become an action potential? I like it. And so how does this become an action potential? We have these small clouds which are depolarizing and hyperpolarizing. And these clouds are driven by the chemical and electrical driving force across that membrane potential. Remember, this is why the ions come in. I just brought this back up to remind you why the ions come in. The sodium was coming in a lot. This shows potassium coming in, but remember, sodium was coming in through the excitatory. And there was a lot of chloride on the outside. Remember, it was the banana in the ocean, so there's a lot of chloride coming in too. There's a good driving force on that. And so what makes it an action potential or not? And so what can happen is we have the dendrites and the soma, and there's something called the trigger zone right at the top of the axon. Another word for it is the hillock, H-I-L-L-O-K. Trigger zone is the hillock. So I've given you a target, remember that. And that'll go in your homework, and you can work on it in section this week. And I like this diagram of an action potential because it shows failed initiations. So you might have the tone played in a fear conditioning experiment that could initiate a lot of glutamatergic inputs and that may or may not drive a fear response. And you could imagine that those glutamatergic inputs are too weak to drive an action potential. So you could have failed initiations of an action potential that's in the right place to drive a fear response. But maybe they're sufficient enough to drive and get an action potential and get this neuron to fire, which could get a fear response underway. And so what makes the difference between failed initiations of an action potential, depolarizations that don't make it, and depolarizations that do make it and cause this violent change in the membrane of a neuron all the way up to plus 40 from minus 70. And basically what changes is that there's a threshold for opening a voltage-gated channel. Remember the finger trap? There's this channel called voltage-gated sodium channel. Here's a big green monster here. Put these green monsters in your drawings for your homework into the axon hillock, okay? And these are, don't get confused because these guys also let in sodium. These are not glutamate receptors. They let in sodium like those glutamate receptors, but they are not gated by glutamate, they're gated by voltage, okay? And so there's this handoff from glutamate letting in a bunch of sodium to now, if enough of those things happen, if enough graded potentials build up, they get the neuron to a threshold, which is minus 55, where you can get sodium through this voltage-gated channel that it's like this finger trap has been pushed together enough to open. At minus 70, it's closed, it's in the membrane, and then it will pop open here and let the sodium in when the voltage across this blue membrane gets to minus 55, okay? And then it can let sodium in. And these things are all along this membrane. There's not just one here, but they are in a chain, so we can have a chain reaction because once one opens, it can help open its neighbor. You don't need the graded potentials anymore. The action potential propagates itself in this all or none phenomenon. It's like a freight train leaving the station from the axon hillock all the way down the axon. And I've got a diagram of that, but just here we have failed initiations, but something that reaches minus 55 lets this voltage-gated channel open. All right, and this, I said they were studded down the membrane of the axon. And so this first one opens. A lot of sodium came in, and that's sufficient to help open the neighbor, which is sufficient to help open the neighbor and down the chain. And this is happening so fast. So tap yourself again. How many, every time you're doing this, you're creating, this axon is getting exercised, right? The whole thing. And your arm is very far, from, just wiggle your fingers too. There's some neurons that are all the way up here in your motor cortex that are controlling especially your fingers. So finger tapping is an even longer axon than this arm tapping. And so you're getting an action potential down every time you're tapping. So this is so fast. And interestingly, it doesn't flow backwards, and we won't talk about it too much, but there's a way, there's an inactivation site at the bottom of the channel to make sure that they close for a little while so that it doesn't just ruin. They set up this great negative resting membrane potential so you're ready for action. And if they just activated each other left and right, both upstream and downstream on the axon, you wouldn't be ready to do the second finger tap. Okay, and so they have to have this brief moment where they're inactive to close everything up again and get ready for the next action potential. So it helps it from not dissipating in two directions. Any questions? So what makes the axon hillock region a trigger zone? Why don't we talk about that? Why is the answer a concentration of voltage-gated sodium channels? Okay, and talk to your neighbor for a minute about this question. Right. Okay, thanks everybody. Let's make sure we get through all the slides. And you'll get to work on this in sections, so that should be helpful. All right, we'll bring everybody back. Thank you. And this back propagation and the inactivation, that's a little bit advanced, so I'm not gonna, if that part is a little bit messy, don't worry, I want you to understand the forward propagation. This is another slide showing you an action potential blown up here. This is just a few milliseconds. It's going from -70 up to about +40, and then it comes back down again. And we've color-coded this action potential here to show you a second distance. Positive ions are rushing in, bringing the axon and the cell 40. So it's very positive, especially in the axon. We're going to be a wave of positivity flowing down the axon. But then it has to get back down to -70. And that is achieved by flow of potassium. And this is kind of unusual for you to see because you've been looking at ions entering the cell a lot. But there's also a channel called a voltage-gated potassium channel. And there's one kind of these that's found here in the axon that allows potassium, remember it's inside the cell, it's positive. Even though it's more negative than positive inside the cell, there was a lot of potassium in there. And that potassium can flow out. And that loss of the positive ion helps bring everything back down to below threshold into that negative potential. Okay, so this is also helping to restore readiness for the next action potential. So you have this action potential that swings out and then it's very positive inside, and then the potassium rushes out, bringing it back down. It can overshoot a little bit, and so you can see it's maybe hyperpolarized. Polarized means how far it is from zero. Depolarizing is going towards zero. Hyperpolarizing is going away from zero into that really negative space. But then it comes back down to rest, and everything's ready to go again. All right, so this is a drawing, and this is a real squid giant axon. This is our first glimpse of the action potential that was seen here. And the conductances were worked out, solutions and things. You could imagine just changing the recipe for what was the squid axon was floating in, and they could work out this one was the flow of sodium, and this part was the flow of potassium. It's very mechanistic.
So the modulation of action potentials isn't just dependent on chlorine for hyperpolarization, it can also be the escape of excess potassium?
Well, it is the sodium. Yeah, we're not talking about chloride at all here. And don't confuse chlorine and chloride. And chloride here was GABAergic. And the graded potentials driven by chloride entry through GABA receptors was something that was offsetting depolarization caused by glutamate binding to glutamate receptors out in the dendrites. And then here we have this voltage-gated conductance called the action potential that's getting started in the axon hillock. And it's involving voltage-gated channels, two kinds. One that's letting sodium in, and one that's letting potassium out in this 1-2 fashion. Okay? And yeah, we'll keep them separate. That's what the homework is going to help you do, is to keep those separate and work those out. And you're going to have sodium as part of your excitatory graded potentials, and you're going to have sodium as part of your action potential, but it's getting in by completely different mechanisms. Okay? And it's causing different outcomes for the cell. They're both sodium coming in these two different ways is helping to generate the action potential, but in one case it's increasing the likelihood of an action potential, and then in the other it's this all or none. It is contributing to the action potential, but it's in these set amounts. This is like a reflex at the knee, you know. It's just something that happens. All right, so finally this often gets short shrift, but we get to the action potential travels in this chain reaction. That excitatory positive depolarizing part of the action potential is traveling down the axon, and it arrives at the terminals where the neurotransmitters are. And once it gets there, the voltage gets positive around these voltage-gated calcium channels. Calcium was also rich outside the cell compared to inside the cell, so it rushes in, and then this calcium helps to cause these vesicles to bind. And a lab member of mine helped out by finding this nice paper where people were able to use a special form of microscopy to get at seeing these tiny little vesicles binding and dumping their neurotransmitters, just as diagrammed here. All right. So we have 3 voltage-gated channels to think about that are important in the axon to think about an action potential. First sodium, then potassium, then calcium. Yes? Yeah, all of these things are important in this interaction, and then the synapses out at the muscle and the muscle fibers themselves are going to have different interactions related to the channels. So yes, yeah, and it would affect their firing or their ability to relax and things like that. Yeah. All right, so we've got the terminals. Remember, these are the ends of the axons, these terminal boutons. And this is where we're going to have these specialized voltage-gated calcium channels coming in as this kind of easily forgotten extra member here of this process, but critically important to get the action potential to happen. And here everyone's like, oh, ions have been moving for the whole hour. It gets a little dry, so let's go back. This you can Let's go back and thinking about failed initiations in the context of the central nucleus, where somebody is in a nightclub and maybe they've had some psychotherapy for extinction learning after they've left the theater of war, and so that maybe they've tried to realize they're in a safe space when they're in Berkeley and going out to a nightclub, and that they shouldn't have a big fear response. And so maybe they have this extinction pathway has been trained up. There's those GABAergic synapses. They did have that initial fear pathway also trained up by being in war. Those would be these glutamatergic synapses from the fear neurons. They're both impinging onto the central nucleus neuron. They hear the loud noise. The noise drives a response on this central nucleus neuron. All 4 of these presynaptic terminals release neurotransmitter. And the amount released will determine whether or not they have failed initiations or they hit the threshold and they get an action potential. Okay, and so who wins? If the GABAergic is larger, then there would be greater hyperpolarization, bringing it away from threshold. And if the glutamatergic was larger, they'd have greater depolarization, bringing it toward threshold. And if this neuron is its action potential is sitting on top of generating that fear response, the fear response could win if these synapses are larger. And maybe the fear response would be properly suppressed by the extinction training, that clinical extinction training that was done, if these GABAergic synapses were larger. And this would be because the graded potentials are offsetting each other at the level of the dendrites. All right, affecting whether or not this neuron gets to threshold or not. Yes? Yes, once you open those voltage-gated sodium channels in the hillock, it's the train leaving the station. Yeah, the action potential is, people have been able to figure out ways to stop it, but it's, for a long time it was thought to be pretty impossible. And so, yes, this is kind of a fight to whether or not it reaches threshold. But if it reaches threshold, it's this all or none event. Yeah, and so we're going to talk about LTP on Thursday. And LTP is the strengthening of glutamatergic synapses is what we'll focus on. And so when synapses get larger, it can make an action potential more likely. But the action potential itself is an all or none event. The action potential doesn't get larger. It's just more likely to happen. Okay, and so these synapses are very large. It makes it more likely that you'll get an action potential. But the action potential isn't bigger if these are bigger. If these are large and they're GABAergic, they make the action potential less likely because more negative ions are coming in. But the action potential isn't any smaller. It's just less likely to happen. Yes? It's this all or none event that goes to +40 and back down. It doesn't, there's no such thing as a larger action potential that would be higher. It would just, it's just that the axon decided to dump vesicles at its terminals or not. This doesn't get any larger. The graded potentials can get larger. More ions can come into the dendrites if these are, if there's larger synapses in the dendrites or more synapses in the dendrites, but the action potential is an all or none event, and it goes up to +40 and back down. It's this clockwork thing. That would be graded potentials, and so that's why you have to keep them separate. In terms of graded potentials can be larger or smaller. The action potential is the same size. Okay? It's just more likely or less likely to happen. There's separate things happening at separate parts of the dendrite. And one's happening in the dendrite, one's happening in the axon. So that's something to work on in section. I know we're running out of time, is think about why the size of the graded potentials can change and why the action potential is always the same size, it's just more or less likely to happen. So that seems like a point we have to work on, okay? All right, thanks everybody. I didn't. I just decided to add it.
Oh, okay, cool.